In this section, we will be discussing lymphocytes. So the topic will be lymphocytes for today. And this is the part of our course, innate immunity. So I'm writing innate immunity right over here. Now let's start with lymphocytes. Lymphocytes are the cells of the immune system. So this is the cell, and that is the lymphocyte I've designed, and they're really important for immune system, specifically adaptive immune system. And from where the word came, lymphocyte, so it is made up of two words, lympho and cytes. Lympho means water. And the second part is cyte. Cyte means cell. Now let's talk about the function of this. Is basically targeted immune response is the main function of lymphocytes. So that's why I'm writing targeted immune response. And types. If we talk about types, so there are different types of the lymphocytes. So if I diagrammatically show you so that you can also understand, let's design cell number 1. So this is, let's say this is our cell 1 because we have multiple lymphocytes. This is our second lymphocyte, and then this is our third lymphocyte. You have 3 major lymphocytes in your immune system. First one is the B cell, B lymphocytes, right? And B lymphocytes, basically, they are the antibody producing lymphocytes, T cells, and they are involved in cell mediate immune system. And then you have next one is null cells, null lymphocytes. They are also known as NK cells that we have covered in the previous lecture also. So if we talk about the origin of this, basically the origin is from the bone marrow, and this is the main cell from where they will be originated. This is lymphoid stem cell precursor. So lymphoid stem cell precursor is the cell that will differentiate into these different cells, right? And bone marrow is where these cells are originated, and basically from the bone marrow, cells will be differentiating from the G0 phase, okay, G0 phase. They remain in the G0 phase, which is resting phase, and cells are not active, they are not dividing, and then they further differentiate into different cell types. Now let's talk about antigen recognition. So let me write it down: antigen recognition and activation. Now, activation requires binding of the antigen. In case of our B cells, there is the receptor called B cell receptor, which is BCR, and that binds with native antigen. It doesn't require any kind of other receptors along with the antigen. So B cell has its own receptor. T cells will have TCR, which is T cell receptor. It requires antigen binding with other receptors. So it will be processed antigen, and you will require MHC molecules for activation of T cells, right? And further, what will happen, then they will undergo mitosis, and also they will undergo clonal expansion. So you get different clones of these cells. So the cells will be targeted against those antigens, and those kind of similar cells will be there. So that's the clonal expansion of the cell. Now, one by one, let's talk about these lymphocytes. Let's talk about B lymphocytes. They are also known as B cells, right? So let me write it down, B cell over here. The maturation and development, if we talk about maturation and development, will happen bone marrow, and then further development happens because of the VDJ recombination, and that contributes for the receptor diversity in B cells. Okay, that's a big topic. Now, if we talk about the receptors that are present on B cells, let me design the cell, and let's design these receptors one by one. The first and far most important receptor in B cell is the BCR, B cell receptor, right? And you know that it's the IgM or IgD antibody that is, IgM and IgD antibody that can bind directly to the native antigen. And then further, you have CD79a. It's also known as Ig alpha, and then you have CD79b also, okay, B also, that is also known as Ig beta. These are really important receptor, and they transmit activation signals inside the cell. Next is CD19 receptor. You have CD21 receptor, they are co-receptors, and then you have CD81 complex. So CD19, CD21, and this is CD81 complex, and all these receptors, they are required for enhanced sensitivity to antigen. Next is MSC class 2 receptor, which is important for processing the antigen to the helper T cells. Okay, so I think now this concludes a lot about structure of the B cell. And if we talk about further, the B cell activation will happen, and then further, lymphoid organs will be there where the maturation and activation of the B cell will happen. Now, further, what happens is after the B cell gets stimulated, it will have really high amount of endoplasmic reticulum. So let me design the endoplasmic reticulum in the B cell. And basically, this is very, very high amount of endoplasmic reticulum, and it is required for protein synthesis because it requires to produce a lot of antibodies, and antibodies, they are proteins, right? And that's why we call them plasma cells. So activated B cells are the plasma cells. They are able to produce a lot of antibodies. So plasma cells over here, they will produce antibodies. And let me design the structure of antibodies so that you can remember that these cells, plasma cells, they can produce antibodies. Antibody is basically a Y-shaped structure. So this is the antibody. And you have heavy chains, you have light chains, and then further it will produce large amount of antibodies, and these antibodies are really important for the neutralization of the antigen, right, and also the pathogens. So first thing that they will do is neutralization of toxin molecules. Suppose these are toxins, and antibodies, these can bind directly to these toxins and then neutralize these toxins. So that is one important part. It is known as toxin neutralization. So let me write it down. Toxin neutralization. Okay, toxin neutralization. That's the function of these antibodies. Okay, so next is neutralization. Next is another important function of these antibodies, where the pathogen, let's suppose this is the pathogen and these are the antigens on the surface of the pathogen. These antibodies can actually directly bind to this pathogen and then cover it up, and that covering is known as opsonization. Opsonization, really important part of our adaptive immunity. So these are the basic functions that are completed by our antibodies. All right, now let's move on to T lymphocytes, really important lymphocytes. Remember, NK cell, null cells, we have already covered in our previous modules, right? So that has already been covered. This is a T lymphocyte. It's also known as T cell. And T cells, they are really, really important. And where the maturation of T cell happens is the thymus gland. That is why they are known as T cells. The maturation will happen, they will mature in thymus gland, and then they circulate in the blood and also in the lymphoid tissue. So that's the overall, all these cells are roaming around in the blood and also in the lymphoid organ, and their role is basically to provide immunity against various pathogens. Now let's talk about the receptors. That's really important. We need to know the structure of the T cell and B cell. First receptor is, again, very, very important, T cell receptor, TCR, important for antigen recognition, right? Next is CD3 complex associated with signaling proteins required for T cell activation. So TCR activation is incomplete without CD3. So that complex is needed. Next is you have co-receptors. One is CD4 receptor, and that binds with MSC class 2. CD4 receptor that binds with MSC class 2 molecules, basically helper T cell. Next is CD8, and that binds to MSC class 1. MSC class 1 proteins, and basically activates cytotoxic T cells. And further, you have co-stimulatory receptor, which is CD28. It is required for the full activation and binds to specifically B7 on antigen presenting cell. Okay, these are the receptors. Now let's talk about antigen recognition and using T cell. So how the antigen is getting recognized using the T cell. So for that, I think let's show it diagrammatically, and let me design the dendritic cell. I hope I'm designing dendritic cell accurately. It's weird structure, but let's see, this is the dendritic cell. Dendritic cell will show the antigen by processing antigen using MSC. This is the antigen, red colored, and that cyan color is the MSC complex. Now, the T cell will come closer to this particular, cell, and then interact by using the receptor that is present on the T cell, and then activation of the T cell will happen. Although you also need multiple cytokines, we'll talk about that in the adaptive immune system also. But you need proper co-stimulation involving various receptors, other receptors, and also some of the cytokines that they are involved in this one. Now, what T cell does, what actually the function of T cell is, they are not involved in antibody production like B cells. So they will not produce any antibodies, right? But they will secrete molecules called cytokines. Cytokines are secreted by these cells, and because of this, they regulate the immune system. The balance of the immune system is regulated, or the immune system in general is regulated by these cytokines. Okay. Now, coming on to more advanced topic, which is subsets of T cells, because you have multiple different types of T cells also. You have major subsets of the T cells where you have CD. Let's talk about the first one. So if I name them one by one, it will be very convenient. CD4 positive T cells, they're also known as helper cells. So they help they help in B cell activation, right? That's why they're called helper T cells. CD8 positive, where the CD8 receptor is present, they are known as cytotoxic T cells because they are involved in killing the infected cells. Third one is regulatory T cells. They are also known as Treg. As you can understand, their role is to regulate the immune system. Next, you have memory T cells that can memorize the interaction that happened with the immune system and antigens, right? So that they can fast initiate the process if there is again the encounter of that particular antigen again. Okay, so now let's talk a little bit more about CD4 positive T cells. CD4 positive T cells, they are also known as helper T cells. They express CD4 receptor and they bind to a particular molecule. I hope now you remember MSC class 2. MSC class 2 proteins. What CD and then subset you have, like you also have Th1 cells. So in helper T cells, you have Th1, you have Th2. Let's talk about Th1 cells. So the first thing that they do is they produce cytokines like INF, interferon gamma, and IL-2. These are important cytokines. They play a very important role in activation of macrophages and also cell-mediated immunity is activated by these cytokines. So helper Th1 cells will play very important role in activation of macrophages, right? And they are also involved in killing the intracellular pathogens. So let's suppose these red dots are intracellular pathogens like bacteria and viruses, and they are basically present inside the cell. So that's an important role of Th1 cells. Now let's talk about Th2 cells. So helper T cells, you have subsets which is Th2 cells. They will produce IL-4, IL-5, and IL-13. These are interleukins, and they are involved in activation of the B cells. Okay, activation or stimulation of the B cells to produce antibodies. Antibodies will not be produced just by B cell. It requires that activation. It requires those cytokines. That is where I told you, right? Interaction requires cytokines. Next is you have Th17 cell, Th17 cells, Th17. So they will produce IL-17 and IL-22. These are the cytokines that are produced, and they are involved in attracting neutrophils, and also they are involved, very much involved in case of inflammation. When inflammation happens, Th17 cells are involved. Now we also have CD positive T cells, cytotoxic T cells, I've told you, right? And it requires MSC antigen. So MSC class 1 antigen will be involved in activating these particular cells, and they are involved in killing the cells. So they will be involved in killing. So that's why I'm writing over here, killing of cells, because they are cytotoxic T cells. We also have another cell. I don't have space. Okay, I'm going to use this space over here. And next cell is Treg, regulatory cells, right? And Tregs, they are very important cells, and they express CD4, CD25, and also FOXP3. And they are involved in suppressing our immune response, and that's why they are important for tolerance. Okay, and further they also produce cytokines IL-10 and TGF-beta, really important cytokines. So you can imagine how complex our immune system is, how different kinds of cells we have, how they play an important role in activation of the immune cell, further how they the antigen, and overall how the entire thing goes. Next is, which I'm not discussing over here, is memory T cells, because you have memory T cells, you have memory B cells. Their role is basically to remember the pathogen. So next time a pathogen comes or antigen comes, immediately they get activated and the immune response is much quicker. So you have effector memory T cells, you have central memory T cells, and if I summarize all those points, so you have CD4 positive T cells, they help and regulate. You have CD8 positive T cells, they basically kill the infected cells. You have Tregs, they're involved in suppressing the immune response. Then you have memory T cells, because they can provide memory, and you can get long lasting memory because of these cells. So I hope we have covered a lot of comprehensive details in this particular lecture. We have covered lymphocytes to a very high extent. Every receptor we have covered, along with their introductory role in case of immune system. So hopefully that you got the enough knowledge regarding the T lymphocytes and B lymphocytes from this particular lecture. And I want you to now you can start reading books, really good books like Immunology by Cube. So that's really good book, and you can start with like microbiology from Prescott and all, and start your journey to actually get more knowledge on this particular subject. Right, so we are still covering our course on innate immunity, and you can imagine how much detail we are covering. So after completing this particular course, that I am confident that if you understood all those points, that you have a very good knowledge of the innate immunity. Now, before we conclude, I have a quick request. If you have watched this video, then please subscribe to our channel, and also you can comment if you like the video, and you can also share the video with your friends, hit the like button. Whatever interaction you can do in this video, please do that, because that helps us create more and more videos, and we feel motivated, and all that effort that we put in our videos, that actually get boosted up when you interact with our content. So please do that. Along with that, I also need to mention that we have the platform called researchgoal.com, where we offer skill-based courses. So right here in my YouTube channel, I create theoretical courses where I try to give all the knowledge without any subscription or anything, so that you get the understanding. In the Research Goal, we provide knowledge and support, which is for specifically for research students who are interested in doing research in the area, biotechnology, microbiology, immunology, and drug discovery in general. We have a lot of courses that will help you understand how to do research, how to actually write research papers, review papers. All that knowledge, you will see that the knowledge you're getting is in a very, very low cost. We are not charging even less than half of the fee that already other course platforms they are taking. And on top of that, many of these courses, they are even many of the modules that we have kept them free for all the students. So you can go and explore and see if you like those videos, because they are also available over there. They are not accessible from the YouTube platform, but I do provide link for those courses, and please go to the platform, support us, so that we can make more videos, more lectures, more courses in our future projects also, right? So thank you so much. I'll meet you in the next one. Then please make some notes, okay? Study hard, and whatever knowledge you'll get is not going to get wasted. So please study sincerely, put your 100% over there, and do not think too much about it. All right, let's move on to the next module.